In a climate-controlled lab outside Kyoto, a team of molecular biologists opened a sealed dish expecting the usual faint shimmer of bioluminescent bacteria. What glowed back at them, according to the draft preprint that leaked in late February, was something the researchers described in cautious language as a pattern of emission that does not correspond to any known luciferase reaction. The cultures were pulsing, not shimmering, pulsing in a rhythm that, when run through signal analysis software, produced a repeating waveform. And when they sequenced the DNA, roughly 11% of the coding region turned out to be synthetic. Lab-made. Inserted. The team hasn't said who inserted it. That's the part of the story that has biologists arguing in private group chats and refusing to comment on the record. Because glowing organisms are not new. Genetically modified glowing organisms are not new. Scientists have been splicing luciferase genes from fireflies into tobacco plants since 1986. You can buy glow-in-the-dark zebrafish at a pet store in Florida. That's settled science. What's not settled, according to several researchers who've seen the leaked data, is how a stretch of engineered DNA ended up inside wild-caught marine organisms pulled from water that's never supposed to have been touched by a pipette. Before we go further, a necessary caveat. A preprint is not a peer-reviewed paper. The leak may be incomplete, misinterpreted, or eventually retracted. The researchers involved have not confirmed the most dramatic claims circulating online. What follows is an attempt to lay out what the documents actually say, what the science around bioluminescence actually supports, and what questions remain genuinely open. Bioluminescence is ancient. It evolved independently at least 40 times across the tree of life, from deep-sea anglerfish to click beetles to a particular species of mushroom that grows on rotting oak in coastal Brazil. The chemistry is almost always the same in principle. A small molecule called luciferin is oxidized by an enzyme called luciferase, and the reaction releases a photon. It's cold light, no heat, extraordinarily efficient compared to a light bulb, which wastes most of its energy as warmth. For decades, the standard view held that every glowing organism on Earth arrived at this trick through ordinary Darwinian selection, and that the genetic toolkit was entirely natural. Then, starting around 2018, something odd began showing up in sequencing data. Researchers cataloging microbial communities in the Pacific began flagging short stretches of DNA that didn't match anything in the reference databases. They looked almost natural. But the codon usage, the order in which certain three-letter genetic instructions appeared, was too clean, too optimized. Biological evolution tends to leave fingerprints of redundancy and compromise. These sequences didn't. They read like code that had been written, not grown. Most of those early flags got filed under contamination and forgotten. Labs handle synthetic DNA constantly. A stray fragment on a pipette tip can end up in a sample. Everybody knew that. So when the Kyoto team began seeing similar anomalies in deep-water plankton collected 300 miles off the coast of Honshu, their first assumption was that their own equipment had introduced the contamination. They recollected. They used a new boat, new sampling gear, sterilized everything twice. The anomaly showed up again. According to the leaked document, the synthetic stretches were not random. They appeared in specific locations within the organism's luciferase genes, not replacing the natural sequence, but supplementing it, as if someone had taken a functioning flashlight and added an extra circuit that made the beam flicker in a very particular pattern. That pattern is where the story gets strange. When the lab timed the pulses across multiple samples, the emission cycle repeated roughly every 1.7 seconds. Within each cycle, the intensity varied in a way that, when plotted, produced a waveform the team's signal processing software interpreted as having what's called low entropy. In plain language, the light wasn't random. It had structure. It looked like information. Before we jump to conclusions, and there are a lot of conclusions circulating right now, it's worth slowing down. Low entropy in a biological signal doesn't automatically mean anything mysterious. Cicadas produce rhythmic signals. Fireflies synchronize their flashes in mangrove forests across Southeast Asia in patterns so orderly they look choreographed. Biology can produce apparent order without any outside engineering. The question isn't whether the organisms are flashing in rhythm. The question is whether the DNA sequence driving the rhythm was actually assembled by human hands, and if so, by whom. The Kyoto team identified 3 candidate explanations in their internal notes. Each has problems. The first is straightforward contamination. Synthetic biology labs around the world produce massive volumes of engineered DNA every year. Fragments escape. A 2021 study estimated that trace amounts of synthetic genetic material can be detected in wastewater downstream of almost every major research city on Earth. It's plausible that bits of engineered DNA drift into the ocean, get picked up by microbes, and eventually integrate into wild genomes through horizontal gene transfer, a process where bacteria and other simple organisms can swap genetic material like trading cards. The problem with this explanation is the specificity. The synthetic inserts in the Kyoto samples weren't random scraps. They were functional. They were in the right genes, and they appeared across organisms that don't normally exchange DNA with each other. The second explanation is a deliberate release. Governments and private companies have been experimenting with environmental release of genetically modified organisms for decades. Mosquitoes engineered to crash malaria populations have been released in the Cayman Islands and parts of Brazil. Modified salmon, modified corn, modified bacteria designed to eat oil spills. It's not impossible that a marine release program, sanctioned or otherwise, resulted in engineered plankton entering the Pacific. If that's the case, a paper trail should exist somewhere. So far, no one has produced it. The third explanation is the one that's driving the online speculation, and that the researchers themselves have explicitly declined to endorse. That explanation proposes the synthetic DNA didn't come from us at all. It's the kind of claim that sells magazines and collapses careers, and the Kyoto team, to their credit, has pushed back on it repeatedly in the limited statements they've released. They describe the sequences as anomalous and unexplained, not as extraterrestrial. There is a meaningful difference. Still, the conversation is happening. And it's happening because the structure of the inserts doesn't fit cleanly into the first 2 boxes either. Dr. Miriam Vance, a molecular ecologist at the University of Washington who has reviewed portions of the leaked data, told a colleague the inserts look, in her words, too clean to be accidental and too weird to be obviously ours. She emphasized that too weird is not a scientific claim. It's an impression formed by someone who has spent 30 years looking at DNA sequences and knows what typical engineering looks like. What she was willing to say on the record is that the codon optimization pattern doesn't match any commercial synthesis platform she's familiar with. That doesn't rule out a private or classified origin. It just means she can't identify the source. Meanwhile, a team in Tromsø, Norway, reported a similar anomaly in deep-sea copepods collected in the Barents Sea back in 2023. Their finding was buried in the supplementary materials of a paper that was mostly about water temperature, and nobody noticed until the Kyoto leak sent researchers scrambling through old data. The Norwegian sequences share, according to preliminary comparison, a roughly 80% similarity to the synthetic inserts found off Japan. 2 locations, 2 hemispheres, roughly the same anomalous sequence. That's when biologists started taking the story seriously. Not everyone is convinced, of course. Dr. Hassan Okoye, a geneticist at Imperial College London, has argued that the apparent match is an artifact of how the sequences were assembled computationally. He points out that both the Kyoto and Tromsø teams used the same open-source assembly pipeline, and that pipeline has known quirks that can make unrelated sequences look more similar than they really are. His critique is serious and deserves consideration. If he's right, the entire story dissolves into a software bug. If he's wrong, something genuinely unusual is circulating through global ocean currents. Independent replication is now underway at 3 universities. Results are expected sometime this summer. The pulsing rhythm is a separate puzzle from the DNA itself. Even if the synthetic inserts turn out to have a mundane origin, the question of why they produce such structured light remains. Some researchers have speculated that the pattern is simply what you get when you add an extra copy of a luciferase gene and let it oscillate against the organism's internal circadian chemistry. A biological accident, in other words. The pulse means nothing. It's just what the math produces when you combine those particular ingredients. Other researchers are less willing to close the door. Dr. Anya Leclerc, a computational biologist who has worked on SETI-adjacent data analysis, ran the waveform through standard signal detection algorithms. She reported that the pattern showed modest but non-zero markers of the kind of structured repetition that information-theoretic models associate with encoded signals. She was careful to add that her analysis was preliminary, that the sample size was small, and that she was not claiming the organisms were transmitting a message. What she was claiming was that the signal wasn't noise, and that further analysis was warranted. Further analysis is exactly what's not happening, at least not publicly. The Kyoto team has gone quiet since the leak. Their lab website is still up, but the preprint has been pulled from the server where it originally appeared, and requests for comment have gone unanswered for over a month. That silence could mean anything. It could mean they're correcting errors. It could mean they're preparing a formal publication. It could mean they've been asked to stand down by someone with the authority to ask. In the absence of information, speculation expands to fill the vacuum. The broader context is worth considering. Human beings have been editing DNA, inserting synthetic sequences, and deliberately engineering organisms for roughly 50 years. In that time, we've released modified organisms into the environment dozens of times, usually with good intentions, sometimes with unintended consequences. The possibility that some of that engineered material has drifted, mutated, and taken up residence in places we didn't plan to put it is not a fringe idea. It's an expected outcome of 50 years of intervention. What would be genuinely surprising is if none of it had escaped. That framing tends to remove some of the drama from the Kyoto findings. Much of what's circulating online treats the synthetic DNA as evidence of something clandestine, either a hidden government program or something far stranger. A more boring and probably more accurate possibility is that we are beginning to detect the slow accumulation of decades of biological tinkering, spreading through ecosystems in ways no one specifically intended. The planet is becoming, in small and scattered ways, a laboratory. Not because someone made it that way on purpose, but because the tools to edit life got cheap and the consequences got loose. If that's what the Kyoto samples represent, the implications are still significant. It would mean that background levels of synthetic DNA in the wild are higher than current regulatory frameworks assume. It would mean environmental monitoring protocols need updating. It would raise questions about long-term ecological effects that nobody has modeled because nobody knew they needed to. If the samples represent something else, something more targeted, the implications are harder to predict. A release program no one has disclosed would be a policy scandal. A signal embedded in engineered life would be, depending on your worldview, either the story of the century or a cautionary tale about pattern matching on noisy data. Most likely, the truth is somewhere between the boring and the remarkable. Most scientific mysteries resolve that way. The phenomenon turns out to be real but has a prosaic explanation once you look closely enough. The pulsing rhythm has a chemical cause. The synthetic DNA traces back to a hatchery experiment from 2011 that nobody remembers. The Norwegian match turns out to be an assembly artifact after all. The story fades into a footnote, and biologists move on. But until the replication studies come back, the door is still open. Samples are moving between labs in insulated containers. Sequencing is happening in multiple countries. Somewhere in the next few months, the data will either collapse the mystery or deepen it. For now, the organisms keep pulsing. Somewhere in the Pacific, 300 miles off a rocky coast, in water colder than most people will ever swim in, something is giving off rhythmic light. Whatever the explanation turns out to be, the fact that we even noticed is the result of a cheap sequencing tool, a restless graduate student, and a willingness to look twice at data that didn't fit. Sometimes that's how real discoveries begin. A signal that shouldn't be there, a pattern that refuses to be noise, and a small group of people, unsure but curious, deciding to follow the light instead of looking away. The story isn't finished. The preprint is still offline. The replications are still running. What happens next will depend less on speculation than on patient, methodical science, the kind that takes months and rarely makes headlines until the work is done. Whatever the final answer turns out to be, the question itself is worth sitting with. Because if even a fraction of what the leaked document suggests holds, up under scrutiny, the line between natural and engineered life is blurrier than most of us have been living as if it is. And that blurring is happening not in some distant future, but in the dark water under a fishing boat, right now, pulsing quietly and waiting to be understood. There is a historical dimension worth remembering here. The first time anyone deliberately transferred a gene from one species to another was in 1973, when Herbert Boyer and Stanley Cohen moved a piece of DNA from one bacterium into another and showed that the recipient expressed the new instruction. That moment, now more than half a century old, set off an industry. Today, the same trick that took two Stanford professors months of painstaking work can be performed by a high school student with a kit that costs a few $100s. The technology has not only matured, it has democratized. That means the number of people capable of inserting synthetic DNA into living organisms has grown from a handful in the early 1970s to, conservatively, millions today. Most of them are working inside well-regulated research programs. Some are not. The escape of engineered material into ecosystems, whether by accident or carelessness, has become more likely every year, not less. What the Kyoto and Tromsø samples may be showing, in the least dramatic interpretation, is the cumulative fingerprint of that democratization. A low, diffuse background of synthetic sequence drifting through the oceans of the planet, occasionally settling into an organism and going along for the ride. If that's the baseline we're moving into, then the question, for regulators, becomes less about whether engineered life can be contained, and more about how to monitor and manage a biosphere that will, going forward, always contain some proportion of human-designed sequence. There is also a more hopeful framing. If synthetic inserts integrate smoothly and stably into wild populations, it means the boundary between the lab and the ecosystem is softer than some worst-case scenarios imagined. It also suggests that if and when we want to use engineered organisms for beneficial environmental purposes, such as breaking down plastics, fixing carbon, or remediating heavy metals, the biology is capable of accepting and maintaining the modifications. The same permeability that allows accidental drift also makes deliberate intervention more feasible. That's double-edged, but it's not only dark. Coming back to the specific case of the glowing organisms, there's one more detail from the leaked report worth including. The pulsing rhythm, according to the researchers' notes, appears to intensify when the organisms are placed under mild stress. Temperature fluctuations, salinity changes, small variations in dissolved oxygen. The pattern becomes sharper, more structured, more distinct from background. Biologists familiar with stress response behavior in bioluminescent species have pointed out that increased luminescence under environmental pressure is itself not unusual. Many natural bioluminescent organisms use light as a startle response or a distress signal. What's unusual in this case is that the structured quality of the signal, its internal rhythm, also appears to tighten under stress, as if whatever is producing the pattern is not just turning up the volume, but also clarifying the message. That could still be a quirk of the chemistry, a particular enzyme responding to environmental change in ways that happen to make the signal look more ordered. But it's the kind of detail that keeps careful researchers looking, because it's the kind of detail that, if it holds up, becomes hard to explain without invoking something more than coincidence. For now, what we have is a story that could go several directions. Mundane, remarkable, somewhere between. The sequencing data exists. The replication studies are running. The researchers are holding their silence until they know what they have. A few months from now, the answer may be clearer. A few years from now, this episode may be a chapter in a textbook on how the 21st century noticed its own biological footprint. Or it may be a chapter in a different textbook entirely. The honest answer right now is that we do not know. What we can say is that something is pulsing. Something carries engineered sequence in its genome. Something produces a rhythm that human signal analysis tools consider non-random. Every one of those statements rests on real measurements, made by real scientists in a real laboratory. The interpretation is contested. The measurements themselves are not. The coming months will determine which of the candidate explanations holds up. The labs running replication studies are some of the most respected in the field, and if the original findings were the result of contamination or instrument error, that will become clear quickly. If the findings hold, the conversation moves to the harder questions about origin, mechanism, and meaning. Either outcome is valuable. Science advances through both kinds of results. Whatever the verdict turns out to be, the researchers who first noticed the anomaly have already done the thing that matters. They looked twice. They asked whether the pattern was real. They refused to let the data get filed under contamination just because contamination was convenient. That kind of attention is the beginning of every genuine discovery, and it's the prerequisite for noticing the ones that matter. The rest is patience.